Gainor and Dr. Hata are both ALK researchers at Mass General Hospital. Dr. Gainor is the director of the Center for Thoracic Cancers and the director of targeted immunotherapy at Mass General. And he's also an assistant professor of medicine at Harvard Medical School. Dr. Hata is a principal investigator at the Mass General Hospital Center for Cancer Research. And he is also a medical oncologist and an assistant professor of medicine at Harvard Medical School. And they have both been working on ALK-positive research for quite a long time and were recipients of one of our 2018 ALK-positive research grants. And to start us off tonight, we'd love to hear from both of you, Dr. Gainor and Dr. Hata, about how you got into oncology, what brought you to oncology, and specifically lung cancer and lung cancer research. So if you could start it off, Dr. Gainor.
Sure. Thanks so much for the introduction, Emily. It's great to be here tonight. It's nice to see so many people on the call, some familiar faces as well. I also want to say, being one of the recipients of one of the ALK-positive awards is particularly meaningful for us as investigators, because it comes from patients. And so that is incredibly inspiring and really makes us work that much harder. So a huge thank you to all you do, and it's really such an amazing community. My road to studying lung cancer, in some ways there was a lot of serendipity, much like many people talk about in medicine. When I was a medical student, I thought I was going to be a surgeon, which for anyone who knows my personality, would never have been a good fit. But it was interesting. I had applied. I was here in Boston. And I applied for a summer research opportunity, and they selected 6 people for it. I was one of the 6. 5 of them got paired with surgeons, and I got paired with a hematologist oncologist. So it was kind of that was my first foray, and it was completely random, but at the same time I knew right away that this is an area that just gelled with all of my interests. And I would say that the next kind of piece of serendipity was when I was a resident and trying to identify mentors. I knew I wanted to do oncology. I thought I was going to be a GI oncologist. And so I started trying to identify mentors, and a couple people just didn't have the right time, or they weren't a good fit. And someone said to me, you know what, I know a great person. This was 2008. I know the best person for you to work with. It's a brand, one of our new attendings, and this was Alice Shaw. So I was a resident. She had just come on staff a year or so earlier, and as a resident, she said, there's this brand new genetic alteration that had just been discovered the year before, and it's called ALK, and this is what happens, and kind of the rest is history. And I decided to do my fellowship training and everything, and that has been a longstanding interest. So yeah, it goes back to 2008.
Thanks. How about you, Aaron?
So hello, everybody. Thanks for having me here tonight. It's really a pleasure to talk to you. So, so my decision to go into oncology, I guess, dates back to medical school. So I was doing my MD PhD training at Vanderbilt, and I had just finished my PhD, which didn't have anything to do with oncology. And I was trying to decide, heading back into medical school, what clinical area I wanted to study. And that was about the time that EGFR mutations were discovered. And so what I knew that I wanted to do was basically to do research in the lab that was going to be easily translated to the clinic. And so, these papers, the papers that describe EGFR mutations were sort of a light bulb went off saying, wow, you can look at patients, you can figure out what's going on, and then you can directly act on it. And so that's sort of when I decided that I wanted to do oncology, because I felt like this was a place where what was happening in the lab was really close to what was happening in the clinic. And so when I came to Boston for residency, I did a residency at the Brigham. I started looking around to figure out who was doing what types of research. And I ended up doing a postdoc in Jeff Engelman's lab, studying EGFR and KRAS, and then in 2016 came on faculty. And so really, the decision to study targeted therapies and to study, ways that we can understand tumors and target them specifically is really sort of what caused me to want to do oncology. And then, I don't think at that time I knew that I was going to end up studying that exactly, but ultimately it worked out that way. So, I think, and it's been really gratifying that, I think as you guys all know, to be in research and then to see those discoveries in the lab really, impacting patients very quickly, I think has been really a rewarding thing for me as a scientist and also as an oncologist.
Well, we're really thankful for what both of you are doing and how quickly things are moving to help us have better treatment options in the future. Justin, you have a couple slides to show us, I think.
Yeah, this is really a launching off point, but I think both Aaron and I are happy to talk about anything. I know from prior sessions, you've already spoken to a number of our colleagues, Dr. Dugogo Jack, Dr. Lin, and Dr. Shaw. So, what I've focused these slides on are a bit of a different angle, focusing on immunotherapy, but we're happy to talk about resistance or anything else that you'd like. But so.
And you all are welcome to put questions in the chat. You can start entering questions for Justin and Aaron, and we will start asking them questions as we go along.
Okay. So, and I'm not going to get into super detailed here, but I thought a few slides really just to orient people. One of the projects that we wanted to highlight tonight was just the work that we're doing on developing cellular therapies for ALK. Now, just taking a step back, many of you may have heard the term of cancer immunotherapy, that is trying to leverage the immune system to attack cancer. Typically we think of the immune system as battling viruses, bacteria, but we know it can also attack cancer. Most of the time, when we're talking about cancer immunotherapy and lung cancer, we're talking on the right side of this slide. So these checkpoint inhibitors, these are drugs like nivolumab or pembrolizumab or Keytruda, not to use brand names too much, but essentially those are drugs that are aimed at blocking the brakes on the immune system. And so these are intravenous medicines. And while those drugs have really been transformative for a small subset of people who don't have targeted therapy options, that they haven't been as successful in patients with ALK or EGFR. And so we need to think of additional strategies. Now, I believe Mark Awad and Roberto Chiarli either have or will be probably speaking to you about their vaccine efforts. And that's something that we're actually working alongside them to do. But tonight I wanted to focus on what's known as cellular therapies. And just to give you a little different schematic of how this varies. And again, some of the details I would ignore, but at the heart of what this is essentially, if we start with a patient, leukapheresis is essentially like giving a blood transfusion or basically donating blood. But instead of just taking out the red blood cells, it actually preferentially takes out the white blood cells. These are like the soldiers of the immune system. So it takes them out of the body. And then the goal is to then somehow genetically engineer them. So you're taking cells that can react to many, many different things. But here, then you're genetically engineering them in a lab. And the goal is to insert something that allows those white blood cells to then recognize something on the cancer. So the goal here would be, could we take white blood cells out of the body, engineer a new kind of receptor on the surface that to enable these cells to then recognize ALK. And then essentially then reinfuse them into the body once this whole process has been done. Okay, so that's the big picture of what cellular therapies are like. And this is not, some pie in the sky. This is something, in certain blood cancers, there are now multiple approved cellular therapies. So this is something that, is used in oncology. It's just we haven't used them as much in solid tumors. But we think that ALK may be an ideal target for this type of therapy. Again, I'm not going to get into too many details here. I just highlight this slide to try to kind of, this is at the heart of what we're trying to do in the lab with Aaron and his team. Is essentially what we're trying to do is sort out the code, sort out the code that T-cell receptor, basically sort out what are the T-cell receptors that can react against ALK. And it's this complicated structure here. I think this is really just to not get too into the nitty gritty, but when our bodies make T cells, the soldiers of the immune system, they basically, it's kind of like a menu. They take one piece from like the red, green, yellow, and blue, and then they kind of combine them all together. And this can create tremendous diversity. And so within your bodies, you have millions upon millions of different T cells. And so it's pulling out what are the unique ones that may be reacting against ALK. And if we can sort out this code, this specific combination of the red, the green, the yellow, and the blue pieces of DNA within T cells, it will help us design this cellular therapy. Because as you can tell from this picture right here, this is not something that you can really design from scratch or just design in the lab. You need to actually find it in a person first in order to then create it and basically clone it so that you can use it in other people too. So essentially, that's what we're trying to do. We are trying to develop a cellular therapy for ALK. And the starting point for us to do that, we're trying to decode what these T-cell receptors are. And the goal is to look at both patients as well as healthy volunteers, as well, to see if we can find these very, very rare T cells and then determine that genetic code. And then that will allow us to develop this as a therapeutic.
So we have a question about whether the T-cell receptor has been identified already. Have you found this yet? Or is this part of, some of our members are already aware of the Enigma Plus project, which is a little bit of a separate story, but wondering whether part of that initiative is to find these T-cell receptors. So maybe you could talk a little bit about what Enigma Plus will be also. And
Yeah. So Enigma Plus, so this it is elucidating novel genomic and immune markers in ALK. It's essentially it's a online protocol that will allow patients anywhere in the United States to participate. This is something where we recognize not everyone's able to travel to Boston and see us in clinic, yet want to participate in research. And so you're right, I view it as very complementary to this effort that I just described. So Enigma Plus, this is something that Aaron, myself, Jessica Lynn, Dr. Dagogo Jack are all involved with. And essentially what it's doing is it will allow patients, if they do consent online, virtually, any archival tissue that is available that they would like to donate, basically for us to study both the genetics of their tumor, as well as the immune landscape. That is, what is the immune system around the tumor? Is it identifying the cancer cells, or are there signals that are being secreted within the tumor environment that are somehow blocking them from actually attacking the cancer? So complementary effort. So for the current, what I just described, what the first step has been is actually there we're relying on blood samples. So we're relying on blood samples, not the typical blood samples you might think about. These are blood samples where we're actually isolating the immune cells from the blood samples. And then Jamie Heather, a very talented postdoctoral fellow in Aaron's lab, is looking for rare immune cells in those blood samples that may be reacting to certain portions of ALK. Maybe I'll hand it over to Aaron, and maybe he can give his perspectives on what that looks like, and how they go about identifying what the hits are versus controlling for things.
Yeah. So, to answer your question first, we have not found the T cell yet, or the T cell receptor yet. So the approach we're using is basically like trying to find a needle in a haystack, or really a needle in a stack of needles. So if you think about it, in our bodies, we have millions and millions of T cells that are recognizing all kinds of different things, especially viruses and bacteria and things like that, our immune systems take care of. And, if you take those T cells and you sequence them, you can see all kinds of different T cell receptors, and you can get their sequences. But one of the challenges is that just from the sequence alone, it's very difficult to know exactly what those T cells are recognizing. So the approach that we're taking is to do a functional test where we take the T cells and then we expose them to pieces of the ALK protein. And then we look to see, can we see T cells reacting to different parts of the ALK protein? And so you can take, millions of T cells and you can expose them to the protein, and then you might see one or two of them that react to it. And so our thought is that if we can find, out of millions of T cells, we can find the one or two that are reacting, and then we can isolate them, and then we can study those. Then we've now narrowed down, from millions of T cells to the few that we think are most likely to be reacting. And so we've been collecting blood, and we've been exposing them to pieces of the ALK protein and looking to see which blood samples have T cells that are reacting. And now we've found a number of different blood samples. And so the next step in what we're currently doing is now taking those T cells that are reacting and starting to sequence those to figure out what the T cell receptors are. And then once we know what the sequence, then we can put them back and test them against lung cancer cells and things like that to really prove to ourselves that they are, in fact, reacting, the ALK protein as it would be presented on a cancer cell. So we're in the middle, where we definitely have blood samples where we can see T cells reacting. We're trying to sequence them to understand what the sequences are, and then we'll be able to do the functional testing to prove to ourselves whether they're reacting to ALK and exactly which part of ALK they're reacting to.
Okay. And are there different reacting T cells in each person, or is it kind of specific per the type of cancer?
Yeah, so this is where the complexity of the immune system, which is an amazing thing and allows all of us to be able to fight off all the viruses, becomes a little bit of a challenge. So, we all have different genes that make up the sort of the immune system and how proteins are presented. And so depending on, we call this HLA type, you may have heard that, but that basically means the genes that the immune system recognizes as normal human, your cells genes that also help the immune system recognize things that are not normal. And so each of us has a different selection of these different HLA genes. And so that does affect which peptides, which parts of the ALK protein would be presented. So one of the challenges is we may need to find different T cells for different patients that have different complements of these HLA genes. And so there are certain HLA genes that are more prevalent in certain ethnic groups. So, some of them are more attractive in terms of if we found a peptide, that an ALK protein or a peptide that was recognized by the immune system, that might be common to more patients. But it is going to be different types of T cell receptors for different patients.
And this is actually where some of the a lot of the research can collide, and how some things that we didn't necessarily think, so 7-8 years ago with all these cell lines that where many of you may have heard of efforts ongoing in Aaron's lab of if there's a biopsy trying to generate a patient-derived cell line. And we've used that to both explore resistance to targeted therapies, as well as thinking about the next targeted therapies and combinations. But that can also be used for this work, in the sense that, whether a T cell reacts against ALK isn't just a yes or no. There can be different strengths of some can react more strongly than others. And so part of the efforts will be not just finding it, but finding the ones that can be used for the broadest number of patients, but also the ones that are strongest reactive, and the ones that also don't have bind to other things. So in part of this, many of you may know that, ALK is very closely related to ROS1. That's why you can use a lot of the same drugs, but we also are testing, can we actually have a T cell just react to ALK and not ROS1 to try to minimize some of the bystander side effects. So that's just an example of where we can then use those same cell lines to also get a sense of how strong these reactions are and how specific and things like that.
Dr. Gainor, we do have some members here that are actually ROS patients because they take medications that are the same as what we used to take for ALK. So with this therapy, if you did get it to work for ALK, you feel like you'd be able to then modify it fairly easily to work for ROS patients as well?
So while ALK and ROS1, where the drugs like crizotinib bind are very similar, but other parts of ROS1 are actually quite different. And I brought up ROS1 because when you use so most T cells, actually all your T cells, usually have controls, on switches and off switches, right, to protect ourselves. So we want them reacting against, when there's a virus or the bacteria, but we also can't have them reacting against normal things in our body, and because that can cause issues. So normal ROS1, is present in other places. And so when you do this genetic engineering of the T cell, you essentially remove a lot of the controls. And so that they're much more prone to attack things. And so it's critical to make it so that they're as specific as possible. So the goal would, at least this first wave, to make it as specific to ALK as we could. But I think this would also be an important proof of principle, right? That we can, in lung cancer, that we can use a cellular therapy. And this could certainly then open us up to expanding to all these different other oncogenic subgroups. So I think it's would be critical to just demonstrate the feasibility of this.
Yeah, and I will say that, while we're screening for ALK protein responses, we're also screening for ROS1 at the same time, because it's not that hard to do multiple in parallel. So, it's possible that we may find T cells that are reactive to the ROS1 protein as well.
I think a question that would be on everyone's mind would be, at this stage, you're doing translational research, meaning it's not totally bench-side, but it's also not in the clinic. What's it going to take? Because I'll kind of let everyone know Dr. Gainor and his team were awarded a $200,000 research award in 2018. But then in 2019, we awarded a $100,000 booster because his research looked to be the most promising of the 3 projects that we started in 2018. So we definitely felt that he was, and that was based on some very detailed reports that were submitted by the 3 researchers that we started in 2018. And so if you'd like to explain to us, Dr. Gainor and Dr. Hata, where you've gone from when you first started the research, where you're up to now, what the booster was for, and what it would take to get this into a clinical trial, what still remains to be done.
Yeah, I can lead with that one. So the proposal itself was really a multi-pronged proposal, and part of it was focused. So the first portions of it were trying to really profile the genetics and the immune microenvironment of ALK-positive lung cancers. And so what we've done now is done genetic sequencing on hundreds of ALK-positive lung cancers to look at not just ALK, but what is the spectrum of ALK mutations, or of mutations outside of ALK. Because I took a slide out, but essentially, in order for your immune system to react against something, it has to be sufficiently different. It has to look foreign. And so what are the other mutations that are present in ALK-positive lung cancers? And so profiling that. What we've found is that we tend to refer to this as total mutational burden or tumor mutational burden. That is, it's a crude measure, but it's like how many mutations are present in a lung cancer. And what we found was that at baseline, so most ALK-positive tumors have a low tumor mutation burden. There are some people, as they're exposed to different TKIs, that tumor mutation burden can actually go up. So we actually have found cases where that goes up, but it looks like they're not always the most immunogenic. That is, the mutations that are most likely to elicit some immune response. What we've also done is, and I think this is very familiar to this community, is that PD-L1, which is another one, so typically we think about 2 big biomarkers guiding immunotherapy, TMB, which I just mentioned, and then PD-L1. That PD-L1 can be elevated very commonly in ALK-positive lung cancers. This is something that is often used to guide oncologists. And when that number is high, many times we will try to use those checkpoint inhibitor drugs or PD-1 inhibitors. What we found is that level is commonly high in ALK-positive cancers, but it's not the same thing. It's in some ways it's a red herring. It can be misleading because it's not accompanied by additional immune cells around it. And so this is actually, I think, one of the big things that we are trying to reinforce to oncologists and patients alike, that you can't just send a PD-L1 test and ignore the gene test or start immunotherapy first. You really need to get the gene testing back and look at the PD-L1 testing results. And so, a lot of what we've done is trying to profile the immune environment around the tumor. And that booster award was really to do so using the most cutting edge tool, which is known as single cell RNA sequencing. So everything we've done to date has really been having a pathologist look at slides and look at immune cells around it. But essentially what single cell RNA sequencing does is it can actually look at the genetics at a single cell resolution. Basically, within a biopsy, profile in incredible depth the different types of immune cells there. And we think this is really important and complements all of the cellular therapy work that Aaron and I have been talking about, because it can help inform the research strategies for other clinical trials. And I think that's critical. So for example, I mentioned that PD-1 drugs by themselves, PD-1 drugs by themselves haven't moved the needle in ALK. And so by getting a better sense of what other immune drugs are there, or other immune cells are there, it can help inform combination strategies.
Okay, thank you. There's a couple of questions here related to the Enigma Plus program. Could you let everyone know when you expect that'll be up and running and how they would go about signing up for it?
Yeah, so this is, it was conditioned. So taking a step back, in order for any research to be done, there are 2 steps that need to be cleared. There's getting approval of the scientific review board, as well as the IRB, the Institutional Review Board. So that protocol has been conditionally approved by the IRB, so it's passed both steps. And right now, the only remaining things are the IRB was reviewing the anything that is patient-facing. So even if us, sending out a tweet or a brochure or web page, it's those last things. So the protocol itself has been approved, and the IRB is just waiting to approve those last things. Those are all done, and so we hope within the next month that it will be completely ready to go and go live.
Terrific.
And I know Dr. Lin has been, is also shooting some videos and things like that to also and get into some granularity as well about details for that.
Oh, we look forward to seeing that. That'll be great. A couple of questions here. Would it be part of your research to test siblings or other healthy family members of ALK patients?
It's a great question. Maybe I'll hand that over to Aaron to talk about and make it a little bit broader about testing healthy volunteers for the cellular therapy work.
Yeah, that's a really interesting question. So when we started this, we didn't know necessarily where we might find ALK reactive T cells. And so we took 2 approaches. One was to look in the blood of normal healthy volunteers. So we can get blood from the blood bank that's been donated, and we can look there. And then we also looked in blood samples from ALK patients. And so far what we found, which is maybe a little bit surprising, but we found more potentially ALK reactive T cells in the blood of normal donors from the blood bank. So we don't know the reason for this, and we could speculate why this might be. And I think part of what we're trying to do is to determine whether or not these are truly ALK-specific reactions, or the T cells are seeing something, or they're seeing the ALK peptide, but really they're designed to react to something else. It just looks sort of like the ALK peptide. At the end of the day, it doesn't really matter if the T cell reacts to an ALK protein. It doesn't really matter if that T cell initially was reacting to something else. If it works, it works. But that the question, I've not actually thought about this, but the idea of testing family members might actually be a really interesting approach, because that could be a way of, those would presumably have similar HLA typing. And since we're finding some of these potential responses of these T cells in patients who don't have cancer, that actually might be a good place to look.
Can you talk a little bit? We have heard already from Dr. Watt and Dr. Chiarle a couple weeks ago. So we've heard about their vaccine work. Are you collaborating with them at all in terms of the peptide identification, or looking for T cells that identify the peptides that they'll use, that they're looking at for using in the vaccine? How are you working together with them in their project?
Yeah, it's a great question, and I think Boston, we're very fortunate, and it's such an amazing environment, and we also recognize that team science is really the way to get results faster. And so yes, we actually certainly are, and what we've done is now a lot of the work that we've done has formed the basis for other kind of grant applications to do the next line of work. And so we are working with both Dr. Watt and Chiarle on that vaccine study, as well as, so for example, one of the key pieces of where there can be synergy in our projects is that Dr. Chiarle, for example, has developed, mouse models with intact immune systems that have ALK-positive tumors. Many of you may know that normally a lot of the studies that are done, mouse studies, are done without an intact immune system. Makes it hard to study an immune therapy then, right? And so this is actually where we're actually working together, where Roberto, we can use that mouse model to also test candidates. And we've already had discussions with him about this, using that mouse model for our candidate cellular therapies. So I think this is really, I think it's, great kind of team science. And so we'll be also participating in the vaccine clinical trial. So on both sides of town, really supporting one another and supporting the research.
Great. People are anxious to know if there is any estimated target date for the beginning of the cellular therapy clinical trial. What still has to happen before that could go into the clinic?
Yeah, so we are working as fast as we possibly can on this. So what actually needs to happen? So as Aaron mentioned, we actually do have a few hits where we're seeing some blood samples reacting to ALK. Now, what we're doing is really honing in, like, how specific is that, and what exactly are the regions of ALK that those are reacting to, and making sure there's no cross-reactivity. So that's the first part. Once we are confident that we're having consistently, so we've done that part. So once we have consistently reactive T cells against ALK and we know the sequence, the next part is to do the genetic sequencing, and then it will then fall into validating it, basically validating it in the lab. So there are still a number of steps. FDA and regulatory agencies, when you are genetically modifying things and then as a drug, there is a lot of scrutiny, and we need to be 100% right. And so that's why we're spending so much time on the preclinical work here. But I recognize, and we want to move this into the clinic as rapidly as we possibly can.
Thank you.
Kind of a theoretical question. Supposing there was a windfall donation of, a large sum of money, and they said that they wanted to donate it to Dr. Gainor's research, how much would that be able to actually accelerate the process? I mean, are you held back by lack of staff? Are you held back by, would you be able to hire extra staff? Would that work to accelerate it? What would it take to actually make it happen quicker?
Yeah, so I'm going to hand that over to Aaron. Just in the lab. So I think we've worked right to. So in terms of patient samples, we're trying to leverage the most cutting edge sequencing technologies to shave off what used to be very laborious ways of trying to sort out that code. But Aaron, what do you think are the current rate limiting steps?
Well, I think, I think in some respects, money can help to accelerate some things, but I think there are, because this is a sequential process where we sort of need to do each step, we're limited a little bit that, we can't put everything in parallel all at the same time. So I think, each step takes a little bit of time, especially to make sure that, we're sure of what we're seeing is real before we move on to the next step. So, I think, to answer your question as best I could, I think, certainly resources helps, but, I think we're going as quickly as we can in terms of, all the steps that we need to do. I think the other thing we haven't really got into is that now that we're at this T-cell discovery step, we have a number of different parallel approaches that we're using. So, one approach is we're collaborating with a company who has developed some technology to be able to do high throughput TCR sequencing, and then to generate libraries that you can then functionally validate them very quickly. So that's one approach. It's a little bit, sort of on the bleeding edge of technology. So we're very hopeful that it pans out, but it's very new. And so we'll see. Then we have some more traditional approaches that have been used in the past, which are more tried and true, but lower throughput. And so if those work, it's very clear what we need to do, but it's a little bit slower. And then we have another, so we do have some parallel approaches that are going in parallel, and we'll see which one yields results the fastest. But yeah, I think, it is, each step does take a little bit of time, and we have to wait to finish that before we can move on to the next step.
Yeah, I would just add that, I do think that some of this initial work, this discovery work, is very labor intensive, in terms of folks in the lab. and so, having more hands on deck, can certainly accelerate how many samples we're screening, how quickly we can actually go through the different peptides. So those are some of our rate limiting steps. And as Aaron mentioned, also using some cutting edge technologies to also save some time on the cloning front. Typically, so for example, that cloning piece, basically sorting out the sequence, used to take years. We're trying to use some technologies that help us sort out. T cells have an alpha and a beta chain, and sorting them out simultaneously is something that we're trying to use these technologies to shave off time.
Are immune cells typically found inside of ALK-positive tumors? And if so, does that have any clinical significance?
Great question. So the answer is yes. There are immune cells within ALK-positive tumors. We've looked at, there are different types of immune cells though. So we've looked and focused a lot on a particular type of T cell called CD8 positive T cells, because these are really the heavy lifters when it comes to effectively killing cancer cells. And we think those are the actors for classical forms of immunotherapy. And it does look like there are fewer, although not zero, CD8 positive T cells in ALK positive lung cancers. What we've done, though, is with this single cell technique, we've looked at all the different types of immune cells. And so we actually see different constellations of immune cells within these tumors. And currently, in fact, I have a meeting tomorrow to review the next wave of data, comparing that to groups of patients who are more reactive to classical immunotherapies, and trying to identify what are the similarities, what are the differences between the two.
Okay.
If we were to wave a magic wand and you'd finished all your research and you'd found, that it worked and you had success and you were ready to go to phase 1 clinical trials, is this something that would be connected with a biotech company to produce the products, or what would you expect to make it get to that stage?
Yeah, so I do think this is going to be something where we will need to actually work with pharma and biotech in order to bring this into clinical practice. Now we've started even working with them on, sorry, by the hospital, so you still so we've started collaborations, as Aaron mentioned, with some of the folks on the sequencing side, but the same companies actually have some ability and interest in making cellular therapies as well. So I do think we will absolutely need to partner with industry in order to actually bring this into the clinic. I also think, broadly speaking, it may be that we actually need to maybe some patients, it's going to be the cellular therapies that are going to be most transformative. Maybe another group of ALK positive patients, it's actually going to be more PD-1 in a combination, such as a vaccine. So I actually don't think it's going to be a one size fits all. I think, and this is why it's so critical to actually work together with folks like Dr. Wad and Chiarli. And it may be that maybe you need a combination of these things all together to actually work.
Great.
In that line, do you think that the T cell therapy could work for patients who have progressed on ALK TKIs and their cancer has become ALK independent?
Yes, because even cancers that have become ALK independent, what we have found is that they still have the ALK fusion there, and that is essentially what we're relying on. Is essentially the immune cells still being able to identify those cancers, even if they're not as dependent on ALK.
Okay, the ALK is still there in the cell, so that it can still be recognized. And do you foresee that this therapy would work for people who have brain metastases? Will these cells get into the brain?
Great question. The short answer is we don't know. In many of the studies of CAR T cells, which is another form of cellular therapy, those were approved a couple of years ago. They actually do see entry of those cells into the brain. And actually MGH just launched the first CAR T cell specifically for patients with central nervous system lymphomas. So, we recognize that it is a vulnerability, that we need these cells to be able to go into the CNS, and, early days, but at least in blood-based cancers, it does look like they can get in there.
Good.
That's great.
And can you talk a little bit about just comparing CAR T cell therapy to this T-cell transfer therapy that you're working on? I know CAR T-cells are in the news a lot, and so sometimes we wonder, could CAR T-cell therapy work for ALK?
Yeah, it's a good question. So CAR T-cells and these engineered TCRs, they're both cellular therapies, but they have a few core distinctions. So CAR T-cells, one, they've had all the brakes removed from them. So like they are like a missile where all the controls have been removed, all the safety controls. But secondly, they can only recognize things on the surface of a cancer cell. Okay, they can't see anything inside. And so that's the critical distinction, that in the setting of an ALK fusion, ALK is inside the cell. And that's why you can't use a CAR T cell, because it's no longer expressed on the surface. So you need to rely on the body's normal machinery. So Aaron was describing that all of our bodies have developed a machinery. We call it HLA, where essentially it takes little pieces of the insides of our cells and shows it to the outside. It's like, this is what's inside me. And it presents it in a very specific way. It's almost like this. It's like, and so that's what we're relying on for engineered TCRs, those little tiny fragments of ALK that get displayed. That's what we're using for these engineered TCRs. Because of that, because it relies on that presentation, it needs to be HLA restricted. Basically, you need to, we all have different, so HLA types. This is when people have like bone marrow transplants and they do matching. It's kind of like that. You need to have the right matching type and pair it with the cellular therapy.
Okay. And I'll have this be the last question because we're getting past 8 o'clock here, and we want you guys to get back to your Sunday nights. So when you talk about needing diverse approaches, different treatments for different people, what other approaches do you think are promising for ALK? Are there other ones that haven't been fully explored yet that you think have potential?
So, Aaron,
I can tell you about some different approaches that we're exploring in the lab. On the theme of immunotherapy, where we know that PD-1 checkpoint inhibitors don't work very well because ALK lung cancers don't have very many mutations. We've been trying to find other ways of trying to trick the immune system into recognizing the cancer. So, seeing the ALK peptide displayed on the surface is one way that we've been talking about. Another way that we've been looking at is to try to trick the immune system into thinking that the cancers are infected by viruses. So, all of our immune systems are incredibly effective at controlling certain viruses like CMV and EBV that we encounter in normal life. And most of us have seen those viruses and, we're immune to them. And those T cells are very, very powerful against those viruses. So one approach we're trying to use is to try to deliver little pieces of protein that look like the virus onto the surface of cancer cells selectively, so that the immune system will think that it's infected, say, for instance, by CMV. And we know that if the immune system can see that, it's going to be very effective because most of our immune systems are very, very good at detecting CMV. So that's one approach. Another approach is to try to harness other cells of the immune system that are separate from T cells, like macrophages. So there are some different therapies that have come into the clinic designed to specifically stimulate macrophages. And so we're looking to see are there ways that we can pair targeted therapies with drugs that stimulate macrophages to try to use macrophages instead of T cells to sort of augment the response to targeted therapies. And then sort of more in the targeted therapy world, we're looking at approaches where we can actually try to slow down the evolution of resistance. So we've found that sometimes genes get turned on that can increase mutation rates in cells. And so we're looking to see if there are ways that we can inhibit those enzymes that cause mutations to try to slow down the ability for cancers to develop new mutations and become resistant. So those are just 3 examples of new types of approaches that right now are more in the basic science arena, but we're hoping that if they look promising, then we could figure out how to bring those into the clinic pretty quickly.
All right. Thank you both so much for your time. It's really been wonderful to hear what's been going on in the lab and how our research funding has helped move the science forward. So at the end of these, we usually unmute everyone so everyone can say a big thank you to you both for all your work. So if you want to go.
Thank you.
Thank you.
Thank you.
Thank you.
Thank you.
Thank
Thank
you.
you.
Thank you.
Thanks a lot.
Thank you.
Thank you.
Thanks.
That was
You guys
great.
can stay on, if you guys could stay on the line for 2 minutes, I have an announcement I need to make. So
Bye,
thank
Dr.
you, doctors.
Gainor. Bye, Dr. Hata.
Thanks so much. This is wonderful.
Bye,
Thanks for all
doctor.
you've done. Okay. Bye.
Bye, guys.
Okay, I just want to share my screen real quick and go over one announcement. Can everyone see that? So on March 28th is a meeting, ALK 101, which I think some people misunderstand. It's not about ALK, it's about our group. And it's a meeting where our founder, Tom Carroll, will be there, opening up our meeting and discussing the vision that he came up with back in 2015 and in 2017, how we started fundraising. And then Rhonda Mexroth will be there talking about the Facebook group. And then Gina will also talk about the what I call the charity end of the group. But it's a very, very, very important meeting. You just saw on February 28th, you saw Wad, you saw Jessica Lynn and Justin Gainor, 3 people that our group is funded for research for our disease. And I think on the 28th, we need to circle up together and understand what this group's all about and how we can participate in raising future funds, number 1, and number 2, how we can give empathy and support to our members. And I want to thank you for everyone who's showed up on all these Zooms, and Gina and Colin and Emily for everything that you guys do. And it is just amazing that we can sit across people from all over the country. I didn't poll everyone of where everyone's at, but it's an amazing, just an amazing event. And for the doctors to spend Sundays with us, the knowledge and information that we're sharing with others, it's great. So anyways, thank you very much for showing up, and we look forward to seeing you. You got a week off. And oh, by the way, the 26th, I forgot about that. That's going to be where we're going to celebrate. Come on the Zoom at 8 o'clock Eastern time. Bring your favorite beverage, if that's a water, if that's a glass of wine. We're going to do breakout rooms, and we're going to get to know each other and toast some of the good things in our lives, and have a lot of gratitude and appreciation for what we're doing for our fellow patients and members. So thank you very much. I'm going to end the Zoom officially, and we'll see you at the happy hour for the cancer happy hour on the 26th, and then please come on the 28th. And Bill Westlake, thank you very much also. See you guys.
Thank you.
Bye.
Bye-bye.